Good morning to everyone. I am Dr.L.INBATHAMIZH. I am Assistant Professor, Department of Biotechnology, Satyabama Institute of Science and Technology, deemed to be University. I welcome you all to this session of lecture on basic concepts of enzymology. There is another series of enzymes called as regulatory enzymes. They are also called as allosteric enzymes. Now, what is the main difference between the non-regulatory and the regulatory enzymes? In regulatory enzymes, they not only have an active site, but also another specific site called as allosteric site or the modifier site. Now, what is the role of this modifier site? Now, this can bind to a modifier which can be an activator or inhibitor. If an activator binds to the modifier site, it brings about the activation of the enzyme. Whereas if an inhibitor binds to the modifier site, it will lead to the inhibition of the enzyme. The best example for this is ATPase, which is aspartate transcarbamylase. For this enzyme, which is involved in pyrimidine synthesis, aspartate and carbamoyl phosphate are the substrates. Whereas if we see ATP, which is adenosine triphosphate, can act as an activator, whereas CTP, which is the cytidine triphosphate, acts as an inhibitor. So these are called as the modifiers which regulate the activity of allosteric enzymes. If we see the kinetics also, it is not like the non-regulatory enzymes. Non-regulatory enzymes usually follow Michaelis-Menten concept, and the plot will be a rectangular hyperbola. Whereas in allosteric enzymes, they follow sigmoidal kinetics, and their kinetics and the plot will be totally different from the non-regulatory enzymes. Now, all these allosteric enzymes, they have different subunits, hence they are oligomeric in nature. And these subunits, they exist in two forms. One is called as the tensed form or the inactive form. The other one is the relaxed form, which is the active form. So only when the tensed form gets converted to relaxed form, the enzyme activity is the best and activation has occurred. But if the relaxed form gets converted to tensed form, it means it is an allosteric inhibition and the enzyme activity is inhibited. Now, based on this, there are two models. One is called as the sequential model, the other one is called as the concerted model. Sequential model was proposed by Koshland. According to sequential model, the enzyme, the units, the subunits in the allosteric enzymes from tensed state to relaxed state can get converted in a stepwise manner. Whereas in concerted model, which was proposed by Monod, the subunits from the tensed to relaxed state will take place simultaneously together. So if there are 4 subunits, all the 4 subunits will get converted from tensed to relaxed state in concerted model. Whereas in sequential model, one by one, each subunit will get converted from tensed to relaxed state. So these were the models proposed. Allosteric enzymes, they can be either homotropic or heterotropic. When I say homotropic, here the substrate and the modifier are the same. When I say heterotropic, the substrate and the modifiers are different. Now, let me tell you about isoenzymes. Isoenzymes are the enzymes which occur in different forms. They have the same molecular weight, but they will differ in their properties. The best example which we can cite for this is lactate dehydrogenase. This enzyme will exist in heart as well as in muscle. So we can locate these enzymes in heart as well as muscle. So when we think about the subunits, they may differ. Now this heart lactate dehydrogenase will predominate when pyruvate is less, whereas the muscle lactate dehydrogenase will predominate when the pyruvate is in excess. And lactate dehydrogenase, we know that these enzymes, they catalyze the conversion of lactate and pyruvate. So that is about isoenzymes, and they have lot of medical values. On the whole, if we think about the application of enzymes, They are not only used for food industries, medical industries, medicinal purposes, but also for the industrial purposes. Not only that, as we started off, the enzymes, they play a vital role in fermentation reactions. Therefore, they find wide application in industries. Not only that, very important, nowadays enzymes play a very significant role in diagnostics and therapeutics. So not only the diagnostic value, the medical value, the therapeutic value, now the technology has improved so much that we are researching more on enzymes and bringing out new, more friendly concepts about enzymes. So the concept of enzymes is vast. Whatever I could convey in this limited period, I would have done that. Thank you for the patient listening, and we'll meet back again with another interesting topic. Thank you.